phospholipid membrane looks like when there's proteins and stuff in it. It's really important because, again, when we go over action potentials of neurons, we're going to be looking at sodium channels and potassium channels that are going to be allowing charge to come through. There's so many other different types of transporters. What are some other transporters that we... Millions of them.
ATP synthase.
ATP synthase.
Ever. I actually found a cool video if we have time. It's like the world's smallest motor. It just like goes around. It's actually moving in the cell membrane. It's awesome.
And like all these, there's tons of signaling molecules that are stuck in the membrane. Sometimes what happens is different cells will come up to other cells that have these proteins stuck in them, and they'll bind to other cells, and that'll signal in the cell for the cell to do something else, like apoptosis, to die, or to keep living, or to perform some other cellular function. And what I think is really interesting, and this is actually a concept that you would never even go over in Bio 20A, and I'm just going to mention it here because I think it's really cool. But we have this small problem of, we looked at all those amino acids and the proteins, and they're all made of things like oxygen, which is charged, like we discussed last time. They're made of things like sulfur, and they have lots of electrons, so they're polar. A lot of these amino acids, the backbone of the amino acids are polar. They're like water. So the inside of the phospholipid membrane is not polar. It's fatty. So we have this dilemma of how in the heck do you get this protein shoved inside of this membrane? Well, the answer to that question, it's kind of my favorite concept in cell biology, is it's a process called protein translocation, where it's literally like, I'm just going to use an analogy for you guys, it's literally like a sewing machine, that the cellular mechanisms come together and just stitch intricately these proteins inside of this membrane, and it is the most beautiful process ever. All right. So, I was not going to originally give this lecture here, but I wanted to touch on a few things in Henry's molecular biology lecture. This is not my area of study, but I think there's a couple of important things. And this stuff that I don't cover in this lecture is not going to be on the midterm. But some stuff that I do touch on in this lecture, might be on there, but it'll be like a small portion. Okay. So he says, wait, I thought this class was about consciousness. What does molecular cell biology have to do with that? Well, I think we went over that last time. So it's important to remember that there's three kind of domains of life. There's bacteria, archaea, and eukarya. We are eukarya. The plant outside is a eukaryotic plant. The bacteria and archaea are different types of microorganisms, and I don't know the exact difference between them, but they're both prokaryotes. The difference between a prokaryote, some small difference, is that they have a cell wall. They don't have a central nucleus like eukaryotes do.
They don't have membrane-bound organelles, and they don't have a membrane-bound nucleus, so they don't have as much complicated things happening, basically.
All right, we're gonna we want to get to the important part here. This is important. I remember when Henry went over this. We don't really think that, about too much about the bacteria inside of us, but, something like the majority of the DNA, which is in our body, is actually microbial DNA. And, the way that our gut and our stomach and our digestive system is intrinsically related to how we're feeling, and we're going to be going over that in the brain-body connection when Austin comes and gives that lecture. We're going to be learning about cortisol and how that relates to the immune system and how everything is related. But it also comes down to really the kind of inner microbial environment inside of our gut is actually related to our mental state. That's we're going to post this lecture, but not going to be on the gym. So, I know what this slide is about. This slide is about the fact that we have more genes than different types of less complex organisms. Now, with that said, every year when we look at the genome, we realize that we actually have less genes than we thought we did the year before. So actually 10 years ago, we thought that we had 100,000 genes in the human genome. And actually, we now think it's more between like 19 to 20,000. This is even outdated. It keeps going down. Now, how is it possible that a mouse, which is on there, has the same amount of genes that we do, and that we are clearly, at least to us, consciously, like, clearly we're a little bit more complex than like a mouse is? Does anyone have a guess?
Mice have been evolved in the same amount of time as humans, or longer, actually.
I think that's a pretty good answer. Yeah.
Well, to just to kind of to the mice perspective, they may be more complex than us. And it's hard for us to know what they're thinking.
Yeah, it's true. Yeah.
Also, it's not the actual genes that make us an individual from another person or from another organism. It's how the genes fire and how they're turned on.
So that's the answer I was looking for. It's so interesting. We can have one gene in our bodies that is essentially one recipe that we were going over that accounts for like hundreds of different proteins. And the reason why is because it matters in which the way that gene was expressed. And I'm not going to go over with you guys like, the exact little mechanism of how transcription works and how alternative splicing works and how the gene is cut into different places. But all you got to know is that we have only 20,000 genes in us, but for each gene there might be 200 different types of proteins that one gene could produce.
I just learned in cell biology the other day that there's this one gene that encodes some one of our neuron, or is part of our brain, and it's one gene and it can encode 38,000 different proteins. It's just like...
So I just said 200, Jess just said 38,000.
Just craziness.
And that's part of, why it is also that we are, I have the same 99% of my genome is identical to Jess's, and yet we're almost completely different. You know what I mean? It's not only because of that 0.1% of our genes that are different, but it's also because of the way in which our genes are being expressed. And this is going to get into another topic, I think, which is next.
So Julian is also going to be talking about this topic in her lecture on the biology of belief, how the way the gene is expressed is impacted by our environmental conditions. So the central dogma is like DNA, RNA, protein, but then the way that that DNA actually expresses itself is connected to the environment. So it's not just like DNA, it's actually the environment is also impacting how the DNA is being expressed through proteins. So that's really important. And then this other topic, are you going to get to epigenetics?
Yeah, I think there's a stall on epigenetics. But let's just wait till we get there.
Sure.
We're going to talk about a really cool concept called epigenetics, which is going to be the perfect transition for us in neuroscience. Okay, so we are eukaryotic organisms. We went over that. A neuron, which is the basic cell of the nervous system. And there's tons of other cells in the brain too called glial cells, which we're also going to go over, is a eukaryotic cell. It has all the same processes that, per se, a skin cell has in different ways. What makes a neuron different from a skin cell? Well, it's what's your name again? Crystal. It's what exact Crystal just said. It's that a neuron is deciding to express different genes than a skin cell is. So a skin cell has the same cookbook as a neuron, okay? But there's different recipes that are being made inside of these cells. And that's the difference between what makes a neuron and what makes a skin cell a skin cell.
And the stem cells, like before it's differentiated into different types of cells.
Right. Okay. So there's different types of organelles. Not super important. This is the cell membrane again. We already went over it. Mitochondria is a really important organelle in the cell. It's actually really interesting because the organelle is where sugar is processed in the body. It's where glycolysis, or the conversion of sugar into energy, into this molecule called adenosine triphosphate, or ATP, occurs. It occurs in this little area right here in mitochondria. And there's an interesting part about the mitochondria that if you replicate a cell and you take the mitochondria out, so we have a cell in a petri dish and we allow it to undergo mitosis, which is the process by which a cell divides, and we take out the mitochondria before that cell goes under division, that cell will turn into 2 cells and there will be no mitochondria inside of it. That means that the replication of mitochondria is independent of that cell. And actually that mitochondria contains its own DNA, which there's a hypothesis that actually stated that one day back in prehistoric times, there was this kind of larger cell that engulfed this smaller cell, which was the mitochondria, which co-evolved. So it's almost like the mitochondria is a separate organism within you.
And it had the power to do cellular respiration, and so just engulfing that cell enabled us to theoretically.
It's like a symbiotic relationship. So these are some other organelles. I think some important ones to point out. One is called the lysosome, which is responsible for degrading proteins. We have to wonder sometimes, proteins can't stay folded forever. Those weak bonds are not like super strong, and sometimes they're going to get a little like fucked up. So we need to do something to those proteins to degrade them, and then those proteins turn back into their original components, back into the amino acids, and then can go through the whole process of transcription and translation, the central dogma of biology, to become proteins again. Ribosomes. Who knows what's special about the ribosome, given where we are right now? No one knows this.
We didn't really say.
Yeah.
It's the smallest organelle and it transports crap throughout the cell.
Okay, that's good.
ATP.